Hello, everyone. Thank you for joining us. Welcome to the Avexia webinar series. Our topic for today is From Diversity to Function, Translating Metagenomics into Patient Care. Today, we have the distinct pleasure of being joined by an exciting guest, Dr. Lahnor Powell, who will be our presenter for this webinar. Dr. Lahnor Powell is a naturopathic doctor specializing in integrative healthcare, gastrointestinal health, women's medicine, and clinical nutrition. She holds a naturopathic medical degree from Southwest College of Naturopathic Medicine and a master's degree in public health from the University of Alabama at Birmingham, with a focus on community health education and health behavior. Dr. Powell also has doula training, enhancing her expertise in supporting women's health throughout pregnancy and postpartum. As a medical education specialist at Genova Diagnostics, she provides educational support to medical professionals, contributes to peer-reviewed research, and speaks internationally on nutrition, hormones, and gastrointestinal health. Without further delay, I will now turn the webinar over to Dr. Powell.
For the learning objectives, my goal really is to just explain metagenomics, which is whole genome sequencing. It goes beyond just identifying which bugs are present. It helps us to also understand the potential function of the various bugs. And so I really want to help explain that technology and explain what information we're able to derive from what is found. And then looking at the Microbiomix report, we're going to talk about ways to interpret these results in relationship to what your patients may be clinically experiencing, and then basically apply these insights into personalizing the dietary and the supplement strategies for your patient that not only impacts GI health, but also impacts immune function, metabolic health, neurological health as well. So what I really want to ground us on is this one key fundamental idea that the gut microbiome is metabolically active. It is not static. And so our gut houses trillions of microorganisms, bacteria, fungi, archaea, proteus, and they form this beautiful ecosystem within our bodies. And the metabolites, it becomes this signaling molecule, two-way highway kind of street, and it can influence the immune system, metabolic health, neurological connections, even our barrier system within our gut. The signatures, it really reflects our environmental inputs like your diet, and it may act as biomarkers for disease risks or various therapeutic responses. So dysbiosis, which is an imbalance or disruption of the microbes, it's linked to a spectrum of diseases from GI disorders to metabolic dysfunction to autoimmunity to neuroinflammation and so much more. Now, one of the biggest questions I get asked often is, should I run the GI effects comprehensive stool profile or your traditional stool testing, or should I move to metagenomics or whole genome sequencing? And the response is quite simple. It just really depends what information do you want to know. So if you're looking at evaluating markers of digestion and absorption, inflammation, infection, dysbiosis, that's where your traditional stool testing will be very critical. It's the best for helping to identify these specific things. It helps us to see if there's a pathogen and to get sensitivity information to know how to treat that pathogen if your patient is showing symptoms of the presence of that bad bug. So when you have symptoms of diarrhea or constipation or bloating or maldigestion, that's when you still want to consider your basic stool testing options like GI effects comprehensive stool profile. Metagenomics, on the other hand, that tells us more about the function, right? The potential to do something. It assesses the complete microbial diversity, the bacteria, the fungi, the archaea, the protists, and it can reveal functional capacity to make various metabolites like LPS and TMA that might contribute to inflammation, cardiovascular health, histamine, short-chain fatty acids, vitamins, GABA that may contribute to your mood health. So it can really just expand your insight in the symptoms that the patient may be displaying and look at what another potential root cause might be, whether it's explained or even unexplained. So with Microbiomix, the whole genome insight into the gut. So it's powered by whole genome shotgun sequencing, which is the gold standard. And we're able to assess over 28,000 microbial species, the bacteria, the fungi, the archaea, the protists. And we use the microbial analysis platform, which is unmatched reference library, and its ability to be very precise and accurate is also amazing. And it integrates seamlessly with Genova Diagnostics testing that we already offer. So beyond who's there, we really not only want to know what is present, but we want to know what can they do, because ultimately that's what's going to impact health and well-being. Again, traditional profiles will focus on the presence and the balance, where when we move into this type of testing, the microbiomics, it's going to focus on the gene pathways to predict the production of bioactive compounds. And you can see the list below here. Now, we are going to touch upon each of these as we move our way throughout the report. But again, the key understanding point is that it links microbiome function to your systemic health. So let's go ahead and walk through the Microbiomix report. So on the front page of the report, you'll notice that we have, the title of the report, Improving Gut Health. And then the next part, it's going to be called the report summary. This is where we extract out of the report the key findings to really help the patient understand and the practitioner to understand what are those things that we should focus on. Now, this report is very unique in that within every aspect or part of the report, we do put therapeutic and or clinical considerations, which is really helpful when trying to navigate the plethora of information that we get from this report. Then we transition into the actual report, looking at the overview. So the gut microbiome score, we're looking at the diversity or the evenness, richness of the bacterial species compared to the healthy cohort. Then we transition into another part of the test called the key insights, which is basically a really easy way to understand, do I have a potential for higher inflammation? Do I have a potential for higher cardiovascular disease risk or methylation complexities? So it just really breaks it down into a very easy to digest way. And then we dig deeper into the details where we look at the composition, the bacteria, archaea, eukaryote, like how much do we have compared to the healthy cohort, the digestion potential, and then lastly, the metabolites, where we're trying to understand, based on the presence of the various bacteria, what are their abilities to make various metabolites. The microbiome profile, so the breakdown of the various phylum within your gut finding, and then species profile, which lists every single bacteria starting in order of abundance, and it lists everyone that was found in that stool sample. So looking at the individual sections, we have the report summary, where again, we pull out the key information that the practitioner and the patient may want to consider working on to help improve their overall balance of the gut. So for example, this first one, we see a pathobiont was identified. And so if the patient has clinical symptoms that aligns with the presence of this pathobiont, we may want to consider treating accordingly. For the gut microbiome score, we want it to be around 75% because that would indicate that we align closer to the healthy cohort and we have a good interaction between the gut and us as a host. But when we see levels, for example, like you see on the screen, where the gut microbiome score is less than 50%, that means that there's some room for improvement. And the idea is that if we address those things found in the key summary pages that we just looked at, then the actual overall interaction of the gut microbiome and the host will improve and hopefully get closer to 75%. For the diversity, we're looking at the evenness and the richness of the different bacteria species. And what's really nice, again, is that we not only discuss what the marker is, but if you look at the write-up below it, it talks about if you have low diversity, what can I do? You can focus on a diet that's rich in plant-based foods, such as your fruits, your veggies, your whole grains, and even your nuts, because these have been shown to increase microbiome diversity. Now, what's unique about this particular report is that you are given a PDF, but there's also a link to an interactive report that can be readily shared to your patients, as the link for the interactive report is HIPAA compliant in that it's not sharing any personalized information or details. But it allows us to, again, look at the various bacteria species that's present. And with the interactive report, you can actually click on that yellow bolded print, and a pop-up menu appears to the right-hand side telling you more about the various bacteria and what has it been clinically linked to, are there any indications, and it just really helps us to understand it a little bit more in depth. So here we have your microbial community asking the question, does your sample have an overly high abundance of any single organism? If the answer is yes, that means that one of the bacteria species occupies 10% or more of the community. This is an interactive community chart where you can see the largest bubble that is going to correspond with the bacteria that in fact occupies the majority of the space or has a higher abundance. Again, the interactive report, you'll see the key insights. We're looking at the ability to produce butyrate, which is a primary fuel source for the gut cells. It tells you if it's a good level, if it's a bad level, and then it tells you if the sample reported a level similar or dissimilar to the healthy cohort, as well as various things we can do to help improve that particular finding. We're also looking at the impact of your gut through inflammation, the microbiome's ability to break fiber down, to break proteins down, to produce branch chain amino acids, contribute to cardiovascular disease, protect your nervous system, prevent kidney stones, the potential to produce strong smelling gas or flatus, and then also contribute to cell replication and or repair. What's really nice about the interactive report, again, is that it also includes this shopping list where you can click on the tell me why, because it'll say, well, you are deficient in XYZ amount of species, and this particular food can help those particular species become more abundant. So it really helps you to figure out, well, why are you telling me to eat this particular food and not another? But then also you can click the exclude food containing button, like gluten, for example. So if you're gluten free, it'll remove all those gluten foods so that you can be confident in the foods that you are looking at are gluten free, and you can download and print that list and then go shopping. Moving into the next section of the report, where it is called Digging Deeper into the Details, we have the sample composition. So the bacteria compares you versus the average or the healthy cohort. Archaea are like the microscopic life forms like your methanogens. Eukaryota, it's going to include your yeast or your fungi plus your parasites. Novel are those bacteria that have yet been identified, as well as viruses, and human DNA. If we detect more than 4% human DNA, it's either because we touched the swab during collection, or there may be inflammation, in which we need to follow up with our healthcare provider to see if there's any inflammatory condition going on and what might be happening. So when it comes to your microbiome's digestion potential, after you eat a meal, the food gets broken down into your stomach and then it moves to your small intestines where the nutrients are absorbed. Now the food components that cannot be absorbed in the small intestines, like your fiber or excessive amounts of protein, make their way down to the large intestine where the gut microbiota transforms these components into a variety of metabolites or byproducts. So when we look at here the word fiber, if we are eating more fiber, then there will be more bacteria in the large bowel to manage that fiber. So having a higher level would be ideal versus if you have a low level, then we need to increase fiber intake to help change that dynamic community. Mucin deals with the mucin degradation or breakdown. If you have high amounts of mucin degradation or breakdown, that mucosal layer might be a little bit too thin. You might be at risk for intestinal permeability or leaky gut. Whereas if our mucin breakdown is low, then that mucosal layer will be too thick. And then we need to target fiber in our diet to help increase the good bacteria that are capable of degrading mucin. And they degrade mucin because they use it as a fuel source to multiply. For protein, if we see high amounts of protein, again, it can either indicate that we're not breaking our proteins down efficiently, or it can indicate that we have a high protein diet and we may need to kind of make some adjustments in our protein fiber consumption. So this is a picture of our support guide for this particular test where it lists each of the markers and then it talks about the clinical relevance and consideration. So this is a really great resource that you can always download. Let's look at the markers one by one. So hexa-LPS, this one is a pro-inflammatory compound created by certain bacterial species. And when it's elevated, it has been observed in individuals with not only diabetes, but also obesity, heart disease, and non-alcoholic fatty liver disease. So when it is elevated, you can see below, it talks about when you eat more saturated fats, hexa-LPS crosses the intestinal barrier and enters into the bloodstream. So the goal would be to decrease or avoid excessive saturated fat intake. And it lists here some common dietary sources such as butter, coconut products, palm oil, cheese, fatty meats, biscuits, cakes, chocolate, and ice cream. We want to reduce that so that hexa-LPS is not crossing the intestinal barrier. For methane production, there are some species that have the ability to make methane. When we have high amounts, we usually see more slower transit time as well as constipation. And if the patient has a specific compilation of symptoms, we may also need to investigate SIBO, which is small intestinal bacteria overgrowth, if their symptoms are worse after they eat and they experience belching or dyspepsia or something like that, then we might also need to follow up with a SIBO test. For trimethylamine production, if it is oxidized in the liver and converted into TMAO, that's where we see greater risk for blood sugar imbalances and blood clotting and inflammation. So for this one, when we have higher amounts, we want to focus on I3C, which is indole 3-carbinol, DIM, as well as cruciferous vegetables to help reduce the amount of TMA being converted into TMAO within the liver. For the ammonia urease production, when this level is high, we do see it with individuals who have impaired gut barrier function and or inflammation in the gut, and so we want to adjust protein consumption within the diet. For the B. fragilis toxin, if this is identified, if the patient is symptomatic, then we may need to go ahead and treat accordingly, and we're looking for symptoms such as diarrhea. If they are asymptomatic, then we can look at how can we change the diet to help create some friendly competition for this particular bug. So that would be more focusing on our healthy bacteria. Beta-glucuronidase, the production. So this is not the actual measurement. This is the ability to produce it based on the bacteria that were identified. So when this one is on the higher side, it can indicate that you have a higher ability to produce it, which could increase the risk of us recirculating toxins, hormones, drugs, carcinogens, things the body was trying to excrete, basically interfering with phase 2 detoxification. So for this one, we want to focus on the prebiotic fibers found in konjac root, which is also called glucomannan, your foods that are rich in calcium D-glucarate as well, such as your citrus fruits, your apples, and other foods as well. For hydrogen sulfide production, So in this case, you have, we see on the screen, higher levels than the comparison group or the healthy cohort. So this is not diagnosing hydrogen sulfide SIBO, although if the patient is symptomatic with the postprandial type symptoms and foul smelling gas and intolerance to high sulfur or garlicky kind of foods, then we may need to investigate and or treat hydrogen sulfide. But this one, we have seen elevated levels with those who have inflammatory bowel disease. And so to help reduce that amount of hydrogen sulfide being produced in the absence of SIBO, we can look at resistant starches, fructooligosaccharides to help reduce those bacteria that have the ability to produce this particular gas. For branched chain amino acid production, higher levels have been associated with metabolic diseases such as obesity and diabetes. And so regulating the levels of the BCAA production is going to be important through not only maximizing muscle mass through regular physical activity, but also looking at dietary intake as well. And then for oxalate consumption, some bacteria have the ability to break oxalates down in the colon. If you have a patient that has high oxalate consumption, then that's ideal. If they have low oxalate consumption and they have a higher risk for kidney stones, oxalate specifically, then we may need to look at dietary modifications where we're limiting the oxalates within the diet. For GABA, if we have low levels have been linked to anxiety and depression. So whether we have low GABA production or high GABA consumption, it could basically result in low GABA overall. And so in those cases, you can see on the screen, we may need to consult with a practitioner that has the ability to work with, mood balance. We may need to look at protein consumption, precursors to your amino acids, precursors to your neurotransmitters, which are your amino acids. We might need to investigate all of that, as well as ask some more questions about your mental health. IPA is a beneficial substance and it's made by the bacteria. It is anti-inflammatory. So it is a really nice antioxidant as well. So for this one, we definitely want it to be on the higher side. When it's on the lower side, we definitely encourage rye, fiber, things that can help increase our ability to produce it. Histamine production. So when this one's elevated, we have a greater risk of high histamine production. It doesn't necessarily mean that we have histamine intolerance, but if we have the symptoms, then we do want to make sure we reduce histamine foods within the diet, or maybe consider DAO, diamine oxidase, which can actually help break down those histamines. For our short chain fatty acids, we're looking at butyrate production, lactate production, propionate production, as well as acetate production. When any of these are on the lower side, again, we can look at the dietary foods listed in the description below to help us understand what do we need to eat to increase the amount of the bacteria that have the ability to produce that particular short chain fatty acid, because these overall have been linked to just better health, wellness, energy production, better mucosal barrier function as well. For the essential vitamins, B12, 9, 7, 2, and vitamin K can be made by the microbiome. But please keep in mind, for example, with B2, only 2.8% comes from the microbiome. So that means that we have to eat the rest. So none of these are going to be 100% production of what the body needs. So we have to make sure that there are dietary sources of these various B vitamins as well as vitamin K. But again, it's going to be important to see how the microbiome might be contributing to higher or better, more efficient levels of these nutrients versus not having enough. Then we move on to the next part of the report where it talks about species of interest. So these are going to be species that have really great literature references regarding if they're high, if they're low, what do, what do we know about it? These are specifically mentioned here. Again, this is a copy of what the interactive report looks like online. And so if you click on the actual bacteria, a pop-up menu does appear on the right-hand side telling us more about that bacteria. Then we get into, it just lists a whole bunch of bacteria. It lists your archaea, like the methanogens, your yeast, fungi, proteus, or the eukaryotes. And then we move into the next part of the profile. So here we can see the breakdown of the individual phylums that were identified in that stool sample. And then we have a species profile. So based on abundance, that's how we create this PDF. We list the bacteria that are highly abundant, and then we just kind of go down the line. And we talk about fuel sources, metabolites produced, metabolites consumed, and the emerging research. So this is what it looks like, the interactive online report, where if you click the yellow font for Bacteroidetes b. dori, you can see the pop-up menu on the right-hand side. It tells you the percentage, and then it just kind of goes through all the information that is listed on the PDF. It's just an easier way to navigate it. You hit the X, the close, and then that part goes away, and then you can click something else. So really, when it comes to what are the key takeaways, it's basically, I would say the main key takeaway is that this is not a static interaction relationship. This is a dynamic relationship with the bacteria that live in our gut, and they can influence our health way beyond just GI health. It can influence cardiovascular and inflammation and autoimmune, just so much that's really critical. So the microbiomics, metagenomic testing, it goes one step deeper than your traditional stool test that can tell you if you have inflammation, digestive issues, and measure your metabolites. This actually tells you the functional potential for these various bugs. So when you combine traditional stool testing with microbiomics testing, you have this really complete, actionable view of what's happening within your patient's GI tract. And using these results can really help you personalize diets. It can help you personalize prebiotics, probiotics, and even supplements for better health outcomes. So again, my name is Dr. Lahnor Powell. I want to thank you so much for spending a little bit of time with me in reviewing why choosing the microbiomics profile. Enjoy.
For any questions or issues, you may contact Avexia Diagnostics by email at info@avexiadiagnostics.com or by phone 5 days a week at extended hours, 8 a.m. to 8 p.m. Eastern Time at 888-852-2723. Thank you, Dr. Powell, for this informative presentation. Thank you again for joining us for this webinar event. Until next time, from everyone at Avexia Diagnostics and Genova Diagnostics, stay healthy, stay safe, and we wish you all the best on your pathway to wellness.